Ora si registrerà la conferenza.
Good afternoon. We are here at the second SEALS Café, which I remind you are two-voice X-ray dialogues organized in a very informal way. So it's like sitting in front of a cafe, having a short discussion among two speakers, one belonging to synchrotron radiation environment. And in this case, I welcome here Veronica Lattanzi from Danish Technological Institute. And another speaker, which comes from an opposite world, has no contact with our society and with synchrotron radiation. And here, let me introduce Maria Laura Giuffrida, a researcher from the Institute of Crystallography, IC-CNR. The two speakers of today, they will discuss about a common theme they have, which is Alzheimer's disease. And they will set up their minds describing this common theme into completely different ways according to their background. So the first speaker will be Maria Laura, and the title of her talk is Alzheimer's disease and the controversial role of amyloid-beta, a look beyond the pathology. And after her, Veronica Lattanzi will speak about amyloid-beta fibril formation as a phase transition from monomer solubility to fibril structure. So let's end up enjoy this talk.
Thank you, Cinzia, for inviting me to this nice event. The last time was very, very interesting, and I hope you can enjoy even this time this virtual coffee break talking about Alzheimer's disease. And thank you all for your time, for being here with us. So let's go to the presentation. Unless the first part of the slides, in which I will summarize very briefly some typical features of Alzheimer's disease. Sorry, the presentation is not in the just, okay. I will summarize very briefly some typical features of Alzheimer's disease. My talk will be focused on the less known role of amyloid-beta as a peptide normally produced during physiology by healthy individuals. I will present to you old and more recently produced data showing the opposite effects of monomers and oligomers to open a discussion together about this under-investigated aspect of the biological role of amyloid-beta, which has been the main topic of my research. And speaking about Alzheimer's disease, we have to start from dementia. Dementia is a range of symptoms associated with an ongoing decline of brain functioning, and it can affect memory, thinking skills, and other mental abilities, sometimes also physical ability. The number of cases of dementia is expected to triple in the next 30 years, and Alzheimer's disease represents 60–80% of the cases. So we can consider the pathology as a global health emergency. The progression of Alzheimer's disease from the first remarkable changes to a clear memory decline is called the Alzheimer's continuum. And we now know that the pathology can start even 30 years before the first sign arrives. During this progression, the brain's overall size starts to shrink. There is a spreading of AD pathology through the affected areas, and in the more recently period, we know that another sign found to progress is the lowering of glucose metabolism that can be assessed by FDG PET. On this continuum, we can distinguish three main phases. A first phase, preclinical and a mild cognitive impairment, and the last, the severe dementia. The length of these phases is not so predictable. it depends on the symptoms of AD. And the three main stages can be also be divided into 5, sometimes referred as 7, as in the case of the table that I reported here, depending on the severity of the symptoms. I reported here a collection of portraits from an American artist, William, an American artist that is passed away in 2007. That is my opinion, and maybe not only in mine, is a clear example of a gradual decay of a mind suffering of Alzheimer's disease. But besides the morphological changes and the arising of symptoms, AD is characterized by morphological changes. The two all main changes, histopathological changes, are the plaques and neurofibrillary tangles. Plaques are extracellular deposits of amyloid-beta, and neurofibrillary tangles are intracellular accumulation of the hyperphosphorylated tau. So within this apparently clear frame, Alzheimer's disease is far from being understood and solved. Today, there are still many open questions, and this afternoon I would like to start with some of these unexplained issues. What we know for sure is that amyloid-beta is produced by its precursor protein APP and is released in the extracellular space. Under pathological condition, it starts to aggregate, as in the scheme that I reported in the slide. The first evidence that A-beta was aggregating fibrillary structure within plaques dates back to 1984, but today there is still a lack of knowledge. In fact, there is no clear correlation between the A-beta plaque and dementia severity. Also, in 19 years, it was published that amyloid-beta not toxic, but to date there is no functions for this peptide, and so the physiological role of the peptide is still far to be elucidated. And finally, the small oligomers that we know are the toxic species responsible for A-beta toxicity has been the target of many A-beta therapies, but today we have no cure. So we have to recognize that there is still a lack of knowledge about the A-beta deposition. So moving to the focus of this talk, but keeping in mind the open question of the last slide, I would like to change the point of view for a while with you, talking about a less known part of the literature in which A-beta is seen as a peptide normally produced by healthy individuals. Due to its early identification within plaques, the biological role of the peptide has been for a long unexplored. But if you consider that the A-beta sequence is well conserved through evolution, and also that A-beta is produced and cleaved through all our life, all of these observations suggest that it's unlikely to be toxic. And I have reported in this slide a timeline from a review that I suggest you as a very interesting reading, in which you can see many publications regarding the physiological role of A-beta. Prior to 2002, A-beta monomers were considered to be simply non-toxic. In 2002 was for the first time reported that it was an antioxidant molecule. In 2003, Plant and colleagues reported that if you deprived mature neurons to a beta cells, you can promote neuronal death. And in 2008 was published one of my favorite papers in which Chirito and colleagues reported that A-beta can be is released into the synaptic cleft, suggesting for the first time that the peptide can have a physiological function involved in the normal neuronal activity. And in these years of change from skepticism to the first recognition of a possible role for the peptide, we too have contributed in this field with for an advancement of knowledge. I would like to share with you our contribution in these fields that is still active and started some years ago in 2009 when we published for the first time a paper regarding the role of amyloid-beta as a peptide neuroprotective. The first problem we faced was the need to have a good starting material that allowed us to have reproducible measurement. So we set up a protocol with the precious collaboration of our chemist colleagues, and the protocol was made up of three steps. One step to promote disaggregation, the second to solubilize the peptide in a proper solution, and finally steps to collect monomers and oligomeric fraction. I reported here some representative approaches that we still use to characterize A-beta when we test its activity. And as you can see from atomic force microscopy, in the monomeric fraction, we found small globule of 1.3 nanometers in 8. While in the oligomeric fraction, we found larger globules of 13 nanometers. And the two solutions showed different activity on cortical neurons, as you can see by the histogram on the right. Sorry. Okay. So we moved on to the biological characterization of amyloid-beta, and what we found was that if you deprive neurons from insulin, thus creating a model of trophic deprivation, you promote neuronal. The presence of monomers prevent to neuronal death, and this preventive activity was blocked by the use of inhibitors like LY294002. That is an inhibitor of the PI3 kinases pathway, a pathway of survival. We also performed a Western blot in which we tested the activity of low concentration of monomers, and what we found was that the monomers in low concentration was able to promote a phosphorylation of AKT, GSK3 beta, and beta-catenin, which are key players in the survival pathway of PI3 kinases. The secret to unmask this function is to use low concentration, far from those used when you would like to promote the aggregation of A-beta. We use nanomolar concentration, which are similar to those found in physiology. And also we search for the receptor engaged by A-beta monomers during its activity. We used a stable clone of fibroblast, stable transfected with human EGF-1R instead of the murine-1. And we performed the phosphorylation in plate using the lysatis of fibroblast, added this lysatis to the plate, pre-coated with an antibody raised again the alpha subunit of EGF-1R. So we captured the receptor on the plate and using a proper buffer and ATP, we search for a phosphorylation mediated by EGF-1 monomers or both together. What we found was that when we treated with A-beta monomers and EGF-1 together, what we found was that monomers was able to potentiate EGF-1 activity. EGF-1 is the natural ligand for the receptor. Okay. And so now going back to the previous scheme I showed you, we can say a little more. A-beta is produced by APP, but now we know that it's not only non-toxic, but it has a function. We demonstrated that A-beta can have a physiologic function that could be lost when the peptide begins to aggregate. And this is not surprising because the human A-beta sequence is shared by 60–70% of vertebrates, and this sequence is well conserved through evolution for 400 million years. So the question is, why should a non-functional and potentially toxic peptide be conserved through evolution? And also the second question that I addressed to Veronica, and all of you deal more than me with the protein structure, is at what stage of aggregation A-beta acquire its toxic properties, and which are the structural changes that occur in the protein just before it begins to aggregate. And coming back to our data, after the demonstration of an interaction between monomers and EGF-1 receptor, we searched for other EGF-1-like activity, like the ability of EGF-1 to promote glucose uptake. We use a non-hydrolyzable sensor, analog of glucose, the 6-NBDG, which is a fluorescent sensor. We use mature neurons deprived from glucose, and we treated our culture with the sensor EGF-1 or monomers. As you can see by the green color of the cells, the presence of EGF-1 and monomers was able to promote the internalization of the sensor, thus suggesting a role of monomers in the glucose uptake. And we also performed an indirect immunofluorescence using an antibody, raised again the N-terminal, the extracellular N-terminal domain of GLUT3. GLUT3 is a glucose transporter located in the synapses, and this is why we use the specific glucose transporter. Okay. This newly found activity should be seen in a more physiological context. So if you remember the work that I mentioned before about the secretion of A-beta during neuronal activity, we tried to mimic this model, looking at glucose uptake during neuronal activity. And to mimic this condition, we used potassium chloride, and we deprived neurons from glucose for 45 minutes, and then we used the sensor, the fluorescent sensor analog to glucose, and the potassium chloride to induce the internalization of glucose. And what we saw was that as potassium chloride do also, monomers was able to promote the internalization of the glucose during neuronal activity. This was very interesting data, because in the context of Alzheimer's disease, we know that the hypometabolism of glucose is an important issue. More recently, we studied the downstream effect of the activation of IGF-1 receptor and PI3 kinases pathway, studying the opposite effect of monomers and oligomers. We treated our neurons with monomers or oligomers, and we look at the phosphorylation of CREB. CREB is a transcription factor important in memory, in learning and memory, that in the recent year has been associated with Alzheimer's disease, because oligomers has reported to decrease the phosphorylation of CREB. We used an increasing concentration of monomers, and what we found was that A-beta was able to promote the phosphorylation of CREB. This was the first evidence of a role of monomers to promote the phosphorylation of this important factor. And to prove the involvement of IGF-1R and PI3 kinase pathway, we used a pharmacological approach using the inhibitor LY-294002, that is the inhibitor of PI3 kinase pathway, and PPP, which is a more selective inhibitor for IGF-1R. As you can see, when we treated our cells with this inhibitor, the effect was blunted. CREB is a nuclear factor, so we performed an indirect immunofluorescence to see if after the treatment the CREB phosphorylated was in the nuclei. And as you can see from the red signal, the treatment with monomers and forskolin, which is a factor that promotes CREB phosphorylation, so is a positive control, we can observe this red signal inside the cells, inside the nuclei. And to be sure of this observed event, we also measured the ability of CREB to bind its target sequence on DNA. In the right, you can see the correspondent signal for the binding to DNA after exposure to monomers or oligomers. As you can see, monomers was able to increase the signal corresponded to the binding to DNA similarly to phoscolin, while oligomers were not able to do it. Okay. And the very downstream effect of the phosphorylation of CREB is the increase of transcript and mature protein of BDNF. As you probably know, BDNF is crucial for neuronal survival, and in the last year has been associated with a deep progression. We now know that aging, Alzheimer's disease, and chronic stress can promote a decrease of transcript and mature protein. So it's interesting. It's interesting to know if the ability of monomers to promote phospho-CREB increasing could be also helpful to increase BDNF, the mature protein. And so we tested by real-time PCR and tested by ELISA the amount of BDNF in conditioned medium. We found a slight increase of the transcript and an increase, a great increase of the mature protein in the conditioned medium. These are the last data I would like to share with you, because even if I don't know if the time is over, but I think it's interesting also in the light of the next presentation, underline that our data are not confined to the synthetic form of A-beta monomers, but we have the same result using native form from conditioned media of overexpressing APP neuroglioma cells. We used the conditioned media to treat our mature neurons, and we had the same effect observed with the synthetic A-beta. It's also important to note that even after 48 hours of incubation, the electrophoretic pattern of A-beta showed a monomeric, showed the peptide in monomeric form. There were no oligomers, and so this is a proof that in a physiological condition, A-beta monomers are stable for a long time. So, in conclusion, and to summarize, we can see that even if Alzheimer's disease is a multifactorial disease with a very complex genetic background, the aggregation of amyloid-beta remains a crucial point. Also, we have to note that until now we have no therapies, and the great part of these therapies are designed against A-beta. All the anti-A-beta therapy developed have failed. So we have demonstrated that A-beta has also a physiological role that can be taken more in account in the future. In fact, I think that the key message that we can underline is that we can have a better understanding of the beta monomer structure in order to understand which changes can drive from physiology to pathology. And these are all the people involved in this story during the last years. I thank each of them and all of you for the attention.
Okay, so sorry if I didn't start in English because my brain was completely. So now if there are questions, curiosities, I think that's a very, very interesting aspect that, at least in my knowledge, was absolutely obscure when I met first Maria Laura, because every time we speak about a disease for Alzheimer, and never in this sense, as she nicely explained to us, that there can be, there is a good side also in this story. So there are some questions left during her talk that maybe the same Veronica would like to address even before her talk. What do you think, Veronica?
Well, I think, well, thank you very much, Maria Lara. It was such a great talk. And I would say that maybe I can move forward with my talk, because maybe a few questions will be answering my slides. And if not, I guess then we can have a general chat all together. And also maybe in the audience, people have curiosities. I think we should go.
So let's complete the two talks, and then we can have a discussion, larger discussion.
Yes, yes.
Okay. So
that's a good idea.
Next, my presentation. Okay.
Now you are the next speaker, Veronica.
Right. Let's hope it doesn't crash. Okay. Sounds good. So, yeah, so as I say, thank you, Maria Laura. It was really nice talk. And of course, thanks to Cinzia Giannini for the nice introduction, and the IC-CNR event organizers to invite me here today. So as you might grasp from the title, I would like to continue this discussion by shifting the focus on the aggregation conditions and fibrous structure of amyloid-beta peptide. And let me know if you see the next slide. Can you? Maybe not.
Click on the presentation.
Yeah. Sounds about what I'm doing. Yeah, okay. Can you?
Yes, okay, fine.
Excellent. So let me start by taking a quick step back, reminding you that we are 8 billion of people in the world, and the decline of infectious diseases via more effective treatments, such as vaccines, for example, increased our life expectancy, and so our aging threshold. But of course, new forms of diseases, as Maria Laura showed before, increased, such as cancer or neurodegenerative disorders. However, other factors can actually contribute to the increases of Alzheimer's disease, and which is a cognitive and behavioral impairment caused by the initial damage of the human brain. And among them, we can list the exposure to polluting factors, sleep deprivation, genetic mutation transferred from the parents to the child, but even the lack of physical exercise, of cigarette smoking, and a poor diet can significantly contribute. So we all should be careful. So unfortunately, the number of cases, as Maria Lara mentioned before, are expecting to triple by 2050, and no effective treatments are available yet, which makes Alzheimer's disease a new silent plague of our modern world to confront with. Okay, so we've seen before that amyloid-beta peptide plays a key role in the development of Alzheimer's disease. So it's present in the brain of healthy individuals. We all have it, and it's produced after the cleavage of an intramembrane protein called APP, which is mainly located in the neuron synapses. So we know that different are the variants of amyloid-beta peptides that are released in the extracellular space of human brains, but this presentation is focused on two main variants, which are amyloid-beta 40, the most abundant one detected in the human CSF, and amyloid-beta 42, which is the main component of the amyloid plaques that are found in Alzheimer patient brains. But now let's narrow this conversation on the peptide. So amyloid-beta is an intrinsically disordered protein, meaning that the single polypeptide chain called monomer doesn't assume a given secondary structure in solution. In fact, it doesn't spiral around into alpha helices or bend and folds into beta strands. Instead, amyloid-beta can fold under given conditions to assume a more ordered conformation when it interacts with other monomers in solution to form small aggregates, known as oligomers, as Maria Laura mentioned before, which gives rise to cellular toxicity. Or in other case, can fold into an elongated three-dimensional fibrous structure that are indeed beta sheet enriched and the main component of those amyloid plaques. So now both the beta 40 and 42, which differ from each other with two amino acid residues at the C-terminus region, can form oligomers or fibrils, and the net charge of the peptide at physiological pH is negative. So now the kinetics and the mechanism underlying this fibril formation has been extensively studied during the past decade. But before we discuss fibril formation, I want you to think of a simple example of a phase transition, here represented by case 1 and case 2, which I'm sure you're familiar with. So when it comes to sugar in water, we will have a general understanding of how sugar concentration and temperature interplay in the phase transition from a liquid phase to a solid phase. But in case 2, we observe that if we add too much sugar, at given temperature, we observe the deposition of a white precipitate at the bottom of our glass. And this means that we exceeded the solubility of sugar in water and the solution will be supersaturated. Now, in similar way, we can describe amyloid-beta aggregation as a phase transition from a monomer phase to a fibril phase when supersaturated conditions are attained. This means that the concentration of free monomers in solution must exceed the solubility concentration before fibrils can separate from the soluble phase and reach a new equilibrium state. So now the development of new effective treatments against Alzheimer's disease require a good understanding of the conditions under which amyloid-beta aggregates form by developing, for example, a method to measure monomer solubility, and what fibrils look like by studying the structure of the fibrils with the new methods, such as small angle scattering techniques. So I would like to highlight that a beta peptide used during my PhD studies were produced as recombinant from E. coli bacteria. So all the samples investigated were prepared after monomerase isolation by size exclusion chromatography, which is a purification step based on a protein separation solution according to size and molecular weight. As an example here, we can observe a general chromatogram of a beta 42 monomers eluting as a clear single peak. So, sign of a pure sample. Okay, so we have seen earlier that amyloid beta variants are released in the extracellular space in the human brain. The hypothesis of a failure in the clearance of the peptide produced in excess, for example, the enzymatic degradation or washed away through the lymphatic system, might increase the local peptide concentration in vivo. So meaning that knowing the solubility concentration of those peptides, it's relevant to better understanding the Alzheimer's disease causes, factors that causes Alzheimer's disease. So approaching the scientific question from a less complex angle, such as in vitro studies, could be one of the future approach to look into the factors that might influence amyloid-beta propensity to aggregate in a more complex environment. So the solubility concentration in vitro is obtained by measuring the concentration of monomers left in solution in the end state of the aggregation process of initial supersaturated monomer sample. And for that purpose, one can combine two methods, represented here by mass spectrometry with an internal isotope label standard and a new microfluid diffusion size technology. Briefly, just to give an idea of the sample preparation, after size exclusion chromatography, a dilution series of A-beta 40 peptides, in this case, with different initial concentration, was prepared in pure buffer. Then the different samples are then incubated under quiescent conditions and a given temperature and time. And via centrifugation, the aggregates are separated from the monomers in solution. Finally, the supernatant sample containing the soluble species is directly investigated with two different methods in parallel. So in order to quantify now the unknown concentration of monomer in solution with mass spectrometry, we needed to add an internal standard with known concentration. So those are accelerated and separated based on the ratio mass charge. And then the sample is digested with a protease and mixed with a method that can absorb the energy from the laser source and use it to ionize those fragments that are the one traveling. Now, both sample of the internal standard and the sample and internal standard, they differ for the molecular mass. So by adding the same concentration of internal standard in all supernatant samples, the comparison of the total intensity of the signal between the sample and the internal standard fragments allowed us to determine the concentration of those monomers in solution. Briefly again, in parallel, a microfluid diffusion size technology was used to measure the concentration for the same series of supernatant samples. And here we see a chip where two streams of liquid are introduced into parallel channels. Those flows next to each other, and one stream is the sample and the other stream is the buffer. Since there is no convective mixing, the only way for A-beta peptide, in this case, is to migrate into the buffer stream is by diffusion. So at the end of the stream, then the streams are split, and the quantity of protein in each stream is determined using an amine-reactive fluorogenic dye, and that interact with amyloid-beta, and the diffusion coefficient of A-beta 40 peptides is in turn used to determine the hydrodynamic radius. So here we combine the data obtained by the two different methods for the supernatant samples collected after 24 hours incubation, for example. On the x-axis, we report the initial total peptide concentration, and on the y-axis, we report the measured supernatant concentration. Yeah, I mean, we can see that the data are fairly in agreement with each other, and this really confirmed the reproducibility of our results. Sample then were incubated at a given temperature, and the supernatant sample were measured at different times. And we confirmed the presence of a metastable zone, which is time dependent and shrink the longer we incubate our samples. Finally, we determined the solubility concentration of 0.36 micromolar for amyloid-beta 40 in pure buffer conditions in vitro, which we know, and Mario Laura might know, is higher than the concentration of beta 40 detected in vivo. So we also identified a stable regime when the concentration of monomers is below the solubility concentration, and in the metastable zone, time dependent, the fibers are not fully formed yet, while in the unstable regime, the aggregation leads to the formation of biphasic state, where fibers and monomer coexist in equilibrium. So the follow-up studies here will be to look into intrinsic and extrinsic factors that can affect the solubility of A-beta 40 in order to mimic the in vivo conditions. We now focus on the fiber structure then. Previous kinetic studies gave insights into the rate constants of the microscopic steps of amyloid-beta aggregation. However, they don't reveal any structural information of the species formed during or at the end of the aggregation. So via secondary nucleation mechanism, we know that fibrils can contribute to the proliferation of toxic oligomers via surface catalysis of the monomers into new aggregates. And here I show that small angle scattering methods combined with solid state NMR structure can provide a detailed model of the fibrils between 1 and 100 nanometer landscape. We see that a fibril is made by filaments. The filament structure obtained by solid state NMR, while small angle scattering provides information about fibril morphology, number of filaments, and cross-section dimension, including the flexible end termini. So a general overview, in small angle scattering experiments, we have a collimated beam of a radiation that impacts the sample and is scattered from the sample with a given angle before being recorded on a two-dimensional detector. And here we present an example of Sachs profile in absolute scale of a sample of a beta 42 fibrils in solution. So we will soon see that this curve can provide us with information on the shape, morphology, and number of filaments in the fibril structure. The Sachs data can be complemented with the scattering profiles at wider angle called WAX, which can give us information about the intramolecular distances in the packing of the fibril structure itself. We observe a sharp peak of 4.7 angstrom, which corresponds to the beta strand separation between the intramolecular beta sheet, while the broad hump at 10 angstrom, it's a little bit more trivial, but it corresponds to a perpendicular distance to the direction of the fibril propagation. Finally, the model calculation on more than one sample of amyloid-beta 42 fibril allowed us to determine the cross-section same-axis dimensions. Here reported as A and B, and the number of filaments otherwise invisible with solid state NMR. So fibril have a cross-section of a near-elliptical cylinder and are made by 2 filaments and 4 monomers per plane, as shown here. The result was reproducible among samples prepared from different proteins batches and for experiments performed at different times. The experimental Sachs profile was additionally combined with solid state NMR structure of the filaments previously published by Colvin and Walter to build the atomistic model of the fibril. So this allows us to observe how the 2 filaments are orienting one to another and which are the amino acids exposed to the solvent and those that belong actually to the fibril core, which are clearly important for the development of site-specific drugs that can inhibit oligomers formation at the surface of the fibrils. We decided afterwards to investigate with Sachs changes in fibril structure after the modification of the primary sequence of the amyloid-beta 42 peptide. So here we can observe that in the presence or in absence of the most common peptide used in the aggregation kinetic studies called thioflavin T, THT, the scattering pattern of the fibril is essentially the same. So a different fibril structure instead is suggested when the setting in position 26 is replaced. Unfortunately, those results can't be shown in this presentation because under journal submission, but we observe, however, discrepancy between fibril structures proposed in the literature between in vivo and in vitro studies. So again, here, future experiments should focus on the factors that affect fibril structure and so the cross-seeding between monomers and fibrils to form new aggregates, which are relevant for the pathology itself. So to summarize, amyloid-beta is an important target in the fight against Alzheimer's disease. The aggregation of amyloid-beta into fibrils can be described as a phase transition from a monomer phase to a fibril phase. Today, we saw together a robust method developed to detect the concentration of one kind of amyloid-beta peptide in solution, and we saw at which concentration this phase transition begins at the chosen experimental conditions. When we think about the fibrils, instead, we took the next step towards understanding their structure by using small angle scattering methods. And in this presentation, we saw that beta 42 fibrils are made by 2 filaments, meaning 4 monomers per plane, and a detailed study on a beta fibril structure in vitro and in vivo could support the design of treatments able to prevent oligomer proliferation, which are linked to the Alzheimer pathology. Now, special thanks goes to my former PhD supervisors and project collaborators, brilliant scientists, and of course, funding agency that supported my research projects. But I want my talk to end with these slides. We have seen in this presentation that small angle scattering methods can be used to answer questions related to incurable human diseases such as Alzheimer's disease. But their application can be versatile and of incredible impact in many different areas of interest in our society. So I've recently joined, as Cinzia previously mentioned, the Danish Technological Institute, which has established a big science center dedicated to mediate between innovative companies and big science facilities all around Europe and the world, such as CERN, DAISY, Max4, and soon ESS, to promote the benefits of using neutrons and X-rays, not only in academia, but also in the private sector. So if you have any additional curiosity, feel free to contact me via mail. So last but not least, thanks to the head of the seminar again, Cinzia Giannini, for inviting me today. Thanks to Candida Giannelli to support us in the setting up of those presentations, and of course to the audience, to the line for your kind attention.
Okay, so there is a question from the audience, from Stefano, and there are two questions left in the PowerPoint of Maria Laura, which I would like to ask Maria Laura if you think that Veronica already replied to you or not.
Of course, I'd like if Veronica can answer to my questions, but also if there are someone in the audience. I think it's a very good point to focus on the structure, on the precise structure that can drive from physiology to pathology in the light of a function for monomers.
I think in the audience there are people who have questions. Shall we see what those questions are about first?
Okay, so Stefano Iotti, please.
I'm having troubles with my...
Okay, may I switch on the camera, or for the privacy rules? I'm not allowed to...
Fine, you can.
Okay, I don't mind.
You allowed us to use your face.
Okay, so good afternoon to everybody, and of course, thank you very much for the two speakers. I have to say that I'm not very expert about Alzheimer's disease, although in the past I performed some in vivo NMR spectroscopy for neurodegenerative disease, but not Alzheimer, because actually the patients are too difficult to study for obvious reason. But I have a curiosity question. Are there in vitro studies about the influence of glucose on the solubility of this beta protein? Because I know from the literature there are some correlation between diabetes, different forms of diabetes and Alzheimer's, and the diet, of course. So my question is, since one of the hypotheses is the different solubility of the different form of proteins, if there are studies performed about the influence of glucose concentration on the structure and the solubility of these proteins. Is it clear, the question?
Okay, yeah, thank you, Stefano, for your question. And I think it's a very interesting question because, you know, studying the solubility of a peptide requires us first to define a robust method, actually, to be able to detect and measure the solubility of those peptides. As I was saying before, we saw that the solubility of a beta-40 peptide in our case is much higher than the solubility, that the concentration of the peptide measured in the CSF in vivo, meaning that other factors might influence and change the solubility of this peptide. So Of course, like as I explained at the very beginning, different factors can affect the solubility, and one of those, of course, is like the disease might be directly connected also to the diet. So it might be that this pathology is intertwined, it is connected to like the diet, and so the glucose intake. And however, I think this will be a very interesting future studies, because in literature so far, many solubility concentrations have been provided. However, the primary sequence of the peptide, if it's not wild type, clearly affect the solubility. Because if we think about a beta 40 and 42, they differ only by 2 amino acids, right? But a beta 42 already is less, much less soluble than a beta 40. And here we are only talking about changes in the primary sequence. So if you add on top of this, if you change the environment, sample environment, and so if you increase the ionic strength of your solution, if you change the temperature, I mean, we're talking about equilibrium, right? So it's a thermodynamic equilibrium. Whatever can affect this equilibrium will, of course, affect the solubility concentration of this peptide. So I think it's a very pertinent question, and it's very interesting, and yeah, so it's a good food for thoughts for future studies.
Yes, I believe I'm a chemist. Once upon a time, I was a chemist. Now
Me too.
Okay. So for sure, the glucose must decrease the solubility of the proteins. It would be quite unseldom that the increasing the glucose concentration increase the solubility of these proteins. I would expect a clear relationship between glucose concentration and decrease in solubility. But I was just curious to know if somebody performed some in vitro study on that. Thank you anyway for the clear answer.
If I can, I would like to answer to your questions, because actually the correlation between diabetes and Alzheimer's disease is based on the resistance of the insulin IGF-1 receptor. And this is another important thing, because I think that our data on the monomeric function is relevant in the field, because the interaction with the IGF-1 receptor can promote its phosphorylation in a condition in which its resistance can occur during Alzheimer's disease. Also, if there is a comorbidity with diabetes. So from a biological point of view, the correlation is based also on the insulin resistance.
Okay, thanks. Thank you very much. And thanks, Tika, for inviting me to the meeting.
Thank you, okay, Stefano. I've seen that there is another question from Francesco Bellotti.
No, Francesco.
Yes, you am.
No. Okay.
Okay, go ahead.
Okay, here I am. First of all, thank you, Maria Laura and Veronica, for your really, really nice presentations. I talk a lot with Maria Laura about Alzheimer's disease, so maybe too much. So I have a question for Veronica for this reason.
I was an additional answer.
Okay. No, very, very nice presentation. I have a question about the starting point. I mean, your expression of amyloid beta. You say that you were overexpressed A beta from E. coli, isn't it?
Yeah.
I was interested in this type of, protocol, and I noticed that I don't know if your protocol is the same. I noticed that when you overexpress the protein, the first residue is the methionine. Mm-hmm. I don't know if you remove the methionine or not. In case not, I suppose, I want to ask you if you have some data that confirm that that your sequence has a more or less the same behavior of the natural sequence that does not contain methionine. Because as you know, methionine can be oxidized during the treatment, and you know that, for example, the natural methionine in the sequence, when is oxidized, changes the aggregation behavior of the peptide. So I want to ask you if you know that the starting methionine residue does not have an effect on the properties of your peptide with respect to the natural one.
Okay,
Thank
so
you.
thank you very much for your question again. So yeah, so basically, the expression purification of amyloid-beta follows the publication was published from Soren Lind's group, and it's called a fuzzy method to purify amyloid-beta. And yeah, so what we did in the lab, I mean, not personally, but colleagues of mine, they checked that the presence and absence of the methionine didn't change the aggregation mechanism, which in our case didn't. So by following the aggregation process of amyloid-beta with and without, there wasn't a major impact on the lag time of fibril formation. And as I say, it's very crucial with this peptide, because short and highly hydrophobic, to have a extreme control on the sample conditions, because anything you do will affect the results in case it's not, your experiment is not performed in pure conditions. If I answer to your question, yeah.
Yeah, sure. Thank you very much. Thank you again.
My chair.
Okay, so I think we are almost there. We scheduled 1 hour time, but if there is any comment, any quick question, any curiosity, you have anyway the possibility to listen again, because it's recorded, this cafe. So, and there are also the email in the presentation, and you can directly write whatever you like to the two speakers. So if no one else has anything else to ask, no, it doesn't seem so.
But I'm really happy. I mean, it seems like there were many people that were interested in participating to this coffee altogether. I think it's a very nice initiative, Cinzia. It's informal, but you know, people have a stage to actually meet with other experts. Because I mean, Maria Laura, she's an expert in bio. I'm not a biologist myself, so I'm a chemist that became a biochemist, physical chemist. So that's great initiative, I think. And so, as Cinzia said, feel free to get in contact with me and Maria Laura, and yeah, and talk about science. We're always very happy to discuss science.
Okay, so perfect. And thank you, Maria Laura. Thank you, Veronica. Thank you all, and see you next time.
Thank you.
Bye.
Thank you.
Bye. Take care. Thank you very much. Bye bye.
Bye.
Ciao.